Hello, everyone. I'm Yun Shu from Department of Microbiology and Immunology, Shantou University Medical College. In the following time, I will share with you the basic knowledge about a well-known infectious agent, human immunodeficiency virus, HIV, which causes a worldwide spreading disease, acquired immunodeficiency syndrome, AIDS. Now, let's begin. Here are the learning objectives. After the lecture, you are expected to 1. Describe the biological properties of HIV, including viral structures, target cell types, replication cycle, etc. 2. List the routes of transmission and risk factors. Explain the pathogenicity of HIV infection and clinical cause of AIDS. 3. Know how to apply the proper lab test for diagnosis and formulate the comprehensive measures for treatment, prevention, and control. The retroviruses, Retroviridae, are enveloped positive stranded RNA viruses with a unique replication cycle. According to the diseases they cause, target cells, viral own shape, and genetic complexity, the human retroviruses are classified into 3 subfamilies. Oncovirinae, including human T lymphotropic viruses, HTLV type 1, type 2, and type 5. Lactin-virinae, including HIV type 1, type 2, and the spumer-virinae. Here, we only discuss the members of HIV family. Okay, first of all, let's take a look at the general properties of HIV. There are 2 species of HIV, HIV type 1 and HIV type 2. They have 40 differences in full-length nucleotide sequence and show some various properties. As compared with HIV type 2, HIV type 1 has relatively high infectivity and virulence, resulting in a global epidemic. HIV type 2 is prevalent mostly in Western Africa, and the disease develops much more slowly. As the picture shows, HIV is a spherical, enveloped RNA virus with a diameter of 80 to 120 nanometers. The envelope is acquired by budding from the plasma membrane and contains viral glycoprotein spikes. The envelope surrounds a capsid. Within the capsid, it contains 2 identical copies of the positive stranded RNA genome. The genome consists of 3 major genes that encode polyproteins. Moreover, viral also contains some enzymes, including reverse transcriptase, protease, and integrase. HIV undergoes a unique replication cycle with the reverse transcription and the synthesis of provirus. Here, we take HIV type 1 as an example to learn about the viral genome. HIV type 1 carries 3 structural genes, GAG, POL, and ENV, and 6 regulatory genes, TAT, REV, NEF, VIF, VPR, and VPU. The 3 structural genes, GAG encodes group-specific antigens, including capsid, matrix, and nucleic acid-binding proteins. POL encodes polymerase, protease, and integrase, and the ENV encodes envelope glycoproteins. three regulatory genes, TAT, REV, and NEF, encode regulatory proteins that involve in controlling expression. 2, VIF and VPU encode proteins required for virion maturation. And 1, VPR encodes a transcriptional activator. At each end of the genome are long terminal repeat LTR sequences. The LTR sequences include promoters, enhancers, and other gene sequences related to binding different cellular transcription factors. Let's look at the viral structure more in detail. On the surface of the virion, could you find lollipop-like spikes? Yes, they are glycoproteins, GP120 and GP41. ENV gene encodes polyprotein GP160. GP120 and GP41 subunits come from the cleavage of GP116. The subunit GP120 is attachment protein responsible for binding to the host cell receptor's CD4 molecule. It can also trigger the production of neutralizing antibody. The GP120 is extensively glycosylated and variable, so that it causes antigenicity drift. These factors impede antibody clearance of the virus and create difficulties for an ideal vaccine development. The subunit GP41 is a fusion protein, which promotes the viral fusion with the cell membrane. As mentioned previously, GAG encodes group-specific antigens, including capsid, matrix, and nucleic acid binding proteins. Within the envelope is the viral matrix, P17, and the core or capsid, P24. P24 has high specificity and it could be used for diagnosis. What's more, there are some other proteins binding to nucleic acid like P7, P9. A PLL gene encodes the three main enzymes that are required for viral life cycle: reverse transcriptase, integrase, and protease. Reverse transcriptase, RT, plays a major role in reverse transcribing a complementary negative stranded DNA, cDNA. RT also acts as a ribonuclease H for degradation of the RNA genome. Lack of proofreading, RT is very error-prone. The error rate for the RT from HIV is 1 error per 2,000 bases. The genetic instability is responsible for promoting the generation of new strains and allows immune escape. The integrase helps a cDNA copy incorporated into the host genome, and the protease enzyme is required to cleave precursor proteins into many viral peptides. Till now, let's review the structure of HIV particle again. Could you identify the structural components of HIV correctly in the schematic diagram? Each virion carries glycoprotein spikes composed of GP120 and GP41. We know GP41 is a fusion molecule. GP120 is attachment protein, interacting with the primary receptor, CD4, and the co-receptor on host cells. The envelope surrounds the viral matrix, P17, and the core or capsid, P24. The HIV genome consists of 2 copies of linear single-stranded RNA, which are associated with reverse transcriptase, protease, and integrase. Thank you for your attention.